All right, good afternoon, everyone. Let's go ahead and start. So it is Saturday, February the 13th, the day before spring break or the week of spring break that starts. So obviously we are not going to have any class this coming Saturday. In other words, no recorded lectures will be sent out. But I did send something in terms of your case studies, guidelines, and your study guide for the forthcoming lecture exam, which is number 2, and that would be held on March the 27th. So that's the Saturday after next one. Hopefully everyone received those. So the case studies, hopefully if you look at the guidelines, that's pretty self-explanatory. They are pretty much like a research paper that you guys have to work on. And that has to do with any famous personality who is publicly known for a disorder that can have to do with any system, basically. Even psychiatric disorders like OCD, bipolar disorder, depression, um, schizophrenia, all of those apply. Or OBGYN conditions like endometriosis, polycystic ovarian syndrome, um, fibroids, and those kinds of things. They all qualify for your case studies. So uh, feel free to choose any one of those, and please adhere to the guidelines that were sent out. If you still have questions, feel free to send me an email, ask me, and I'll be more than happy to explain uh, anything having to do with those. And the study guide, we got a good head start as far as those are concerned, so we still have a couple of weeks to go. So use that to the best of your advantage. I have updated all your grades as of now, both for your lecture and lab exams and quizzes and everything so far. Hopefully you're also keeping up with your lecture quizzes on the Connect website. So some things to keep in mind. All right, so with all that, let's go ahead and get back to our topic of discussion, which was the immune system. We are on chapter 22, as we can see here. And so far, we have talked about how we have basically two different types of immune systems, okay? Uh, and they're not mutually exclusive, of course, they work hand in hand together. So those two are the innate or the inborn or the non-specific, non-adaptive immune system. That includes things like your skin, first and foremost, then the macrophages, the neutrophils, eosinophils, basophils, right? Your granulocytes, that is the mucus that is secreted by your mucous membranes, tears, saliva, even breast milk. All of those body secretions have an antibody called IgA, and they're all part of your innate immune system. So basically, almost every human being is born with that, right? And it provides immunity to most of the pathogens. Uh, so now for today's session, we are basically going to focus more on the adaptive or the specific or the special immune system. And why is it called all of those things? Because we are not technically born with immunity to all of myriad of viruses and bacteria and pathogens that are out in our environment. So we gradually develop immunity against all of those agents with time and exposure. And that is something called active immunity, okay? And that's why you get vaccines as well. Vaccination, all it does is it puts a dead or a weakened version of that virus into your bloodstream. So then your immune cells have an opportunity to come across those pathogens and mount, launch a counterattack. And in terms of antibodies, of course. And once you have a critical mass of antibodies reached in your system, that's when you're immune to that disease. So we keep talking about herd immunity. I'm sure everyone has heard that terms many times over the past year or so, right? Herd immunity against COVID-19 as an example. So what we are talking about here is when a certain number of people, a part of the population, percentage of the population has developed active immunity against that disease, either because of falling sick with it, exposure, or getting vaccinated. Those are the two ways that you get active immunity, okay? And so we are still some ways away from that situation, but in the meantime, there's many things we all can do to keep our immune systems in full functional order, such as the same mantra that I've been saying over and over again. A healthy diet, right? Watch the amount of sugar and salt and saturated fat that you're consuming in your diet. Go for more for plant-based diets, more of a Mediterranean diet, right? So olive oil, plants, vegetables, fruits, that is what you should be emphasizing with minimal amounts of animal-derived foods. Stay away from fatty, fried, fast food type foods, okay? Because that's going to mess up the immune system. And then exercise. So these are it's a two-pronged strategy for strengthening your immune system. So eating healthy and then exercising, be physically active. If nothing else, like brisk walking, at least anywhere from 15 to 30 minutes on a daily basis, most days of the week, that would be excellent. All right. So that gives you cardio workout. Being out in nature and being physically active also boosts your immune system. Getting enough sleep, 7 to 8 hours of recommended sleep. And then, of course, uh, stress management. Try to keep your stress levels down. So doing yoga, meditation, swimming, taking a bubble bath, listening to music, anything that makes your heart rate and blood pressure go down is good in those um, circumstances. All right, so here we are looking at the adaptive immunity or the specific immunity, okay? And so as you can see here, it takes longer to develop than your innate immunity because we are not born with it. We acquire this immunity uh, after exposure over our lifetime to different pathogens. And as I mentioned, I can tell you a lot about who you are just based on your immune profile. What types of antibodies have you developed against certain pathogens? That'll tell me where you were born in a tropical country where there's a high risk of, let's say, tuberculosis infection, TB, not all that common here, or hepatitis B or hepatitis C infections, um, uh, tapeworm infestations. They're way more common in tropical areas. So if you have antibodies against those, then I can fairly accurately predict that you have some part of your life that was spent in those places, okay? So when you talk about the adaptive immunity, as you can see here, it's divided into two types, cell-mediated and humoral immunity, okay? The difference, cell-mediated immunity involves T cells, and they're called T lymphocytes or T cells because they mature in your thymus. Thymus is this endocrine gland in your chest region, in the mediastinum. And then the humoral immunity is so-called because the word humor in ancient times meant liquid or fluid. So these include your antibodies. And your antibodies are derived from what are called plasma cells, which in turn are derived from B lymphocytes. Okay, so the T and the B cells are the two cell types of your adaptive immune system. And so here is a nice little picture to familiarize you all with the with those two types of cells, okay? So here's the cell-mediated immunity, as you can see, T lymphocytes, and then the humoral immunity, which is B lymphocytes, as you can see on the other hand. So now further divisions can be seen here too. So the cell-mediated immunity is further made up of what are called cytotoxic T cells, also called CD8 positive cells. And then the helper T cells, also called CD4 positive cells, okay? So CD8 for cytotoxic and CD4 for helper T cells. All right. And the helper T cells basically are the coordinators and activators of the rest of the immune system. Okay. Because they secrete some chemicals, cytokines called interleukins, as you can see here. And these interleukins are responsible for activating the rest of your immune system. So therefore, if your helper T cells are inactivated, made dysfunctional for whatever reason, the rest of your immune system falls apart. And that is why HIV, human immunodeficiency virus, that causes the dreaded disease AIDS, okay? Basically, HIV targets your helper T cells specifically. So that's a very cunning strategy on part of the virus, okay? Take out the chief coordinator of your immune system and the rest of the immune system just falls apart. That's exactly what HIV does. So that's what you're looking at here. All right. So what do the CD8 positive or the cytotoxic cells do? They do exactly what the name suggests, cytotoxic. They're toxic to other disease-causing cells. For example, what you're seeing here um, is a pathogen of some sort. So your T cell has basically destroyed it, uh, something called apoptosis, which means programmed cell death. Your cytotoxic T cell has literally disabled and digested away the pathogen here by means of apoptosis. Okay, let's move on to the other side. What does the humoral immunity do? It gives rise to specializes into what are called plasma cells, and then the plasma cells further specialize into antibodies, okay? And antibodies are these Y-shaped proteins, as you can see here, also called immunoglobulins, that um, basically neutralize different viruses and bacteria, okay? So all these pathogens, viruses and bacteria that I've been talking about, they're also called antigens, as you can see here, all right? So an antigen is any substance that can bind to either a T cell or to an antibody, okay? It's usually a protein or a large polysaccharide, like a large carbohydrate containing substance. Viruses, bacteria, fungi, all of these pathogens that we have talked about, even pollen, right? Pollen, which are like plant sperm, that's what pollen are. They can act as antigens too. Snake venom is a type of antigen, okay? Um, Bee venom, wasp venom, fire ants, all of those are considered antigens. Anything that can call your immune system into action, any agent that can do that is called an antigen. Even tumors, right? Cancer cells, mutated body cells, they can act as antigens too and activate your specific immune system, okay? So you have all types of antigens. You have both foreign antigens, stuff from outside, as well as self-antigens. So your own body cells can act as antigens as well. But for most of us, thankfully, our immune cells recognize our own body tissues as friendly, not as foreign antigens. But in some people, what happens is that our immune cells recognize wrongfully our own body cells as the enemy. So our own immune system turns against us and starts destroying our own body tissue. And that's when you develop an autoimmune disorder, as you can see here. Autoimmune disorder is your immune cells self-attacking, attacking and destroying your own body cells. Autoimmune disorders are way more common in females, first up, okay? So autoimmune disorders, some of the risk factors for those uh, are being female, okay? Number one. Number two, genetics, okay? So if you have other family members, first degree blood relatives who have autoimmune disorders like lupus or SLE, systemic lupus erythematosus, or rheumatoid arthritis, or hypothyroidism, autoimmune hypothyroidism, these conditions, and you have the genes from the family, that puts you at high risk for autoimmune disorders, right? What else? Uh, Exposure to certain chemicals, right? So continuous exposure to uh, pollution, pollutants, and antigens can also trigger antigens, viruses, certain bacteria, right? They can also trigger your immune system to the point where it self-attacks, it starts attacking its own body. And then, of course, advancing age itself, because the older you get, the more exposure that you have suffered to pollutants, viruses, and so on and so forth. Also, being obese or overweight is a risk factor, because remember what I said about fat tissue itself, it's inflammatory. So the greater the amount of body fat that you have, more obese or overweight you are, the greater the chances that you have your immune system basically acting in all kinds of wacky ways and attacking itself at times, okay? All right, so what else can trigger an autoimmune response? Stress in some susceptible people, especially if you have the genetics for it. Stress, chronic stress can bring about an autoimmune reaction, okay? So these are some of the factors which are often responsible for autoimmune disorders. If I were to choose one of them, the prime one, it's your genetics, it's the family that you come from, okay? All right, so moving on here, antigens also called immunogens because they call your immune cells into action, okay? So a special type of antigens is what you call haptens. What are haptens? These are antigens that are way too small to cause an immune response by themselves, but they have to be attached to a larger carrier molecule in order to be recognized as an antigen, okay? And examples of those would be things like pollen, as you can see here, poison ivy. So the chemicals from the toxin in poison ivy is also considered a hapten. It's way too small, but it has to bind to one of your carrier molecules, which is usually a protein, a blood protein, in order to cause a full-blown um, allergic reaction. Penicillin is yet another example of hapten, okay? So look at this partial list of some disorders which are considered autoimmune. These happen because your immune system self-attacks. It attacks its own body, okay? Look at that, rheumatic heart disease, type 1 diabetes, who would have thought? Even diabetes, which is an endocrine disorder, a problem with your blood sugar regulation, is technically it's an autoimmune disorder. So diabetes is also common in these people, right? People who have the genetics for it, advancing age, being obese, overweight, stress, all of those cause these kinds of autoimmune reactions, right? All right, so let's delve a little bit deeper into the T and the B lymphocytes, as you can see here, okay? So you have what are called the T cell receptors, okay? And T cell receptor is that part of your T cells that can actually bind to an antigen, bind to a pathogen, okay? So that's the T cell receptor. Similarly, you have BCRs or B cell receptors, which bind the antigen or the pathogen to the B lymphocytes, all right? So the two types of them. All right, so let's take a look at some differences between B and T cells. Okay, where are all of them made? Without exception, they're all made in your bone marrow, in your red bone marrow. That is one thing to remember, all right? So all immune cells, your red blood cells as well, come from your red bone marrow. So here's the difference. The B lymphocytes, they actually make direct contact with the antigen. It's like physical combat, all right? Soldier to soldier, body to body. While the T lymphocytes, on the other hand, do not make physical direct contact with the antigen. Rather, they recognize antigens when properly processed and presented to them by some other cell. Okay, so you can say that the T lymphocytes are a little bit more prissy or picky, right? They don't make physical contact with the pathogen or the antigen. They would like some other cells to process the antigen first and then present to the T cell. Okay, so what are some of the subtypes of the T lymphocytes? Those two that I mentioned before, the helper T cells and the cytotoxic T cells, right? Also, helper T cells, also called CD4 positive cells, cytotoxic cells, also called CD8 positive cells. Here's the difference between those two types. Here are the T cells you can see, okay? The helper T cells, that is, the CD4 positives, okay, here. And these ones are the cytotoxic or the CD8 positive cells, those two types of T cells. And here is a B cell, the one that will eventually give rise to antibodies from the B cells here. All right, so what I just mentioned was T cells are a little prissy. They like other cells to process the antigen and then present them for further processing to the T cells. So what types of cells act as these go-betweens that will take up the antigen, process it properly, and then present them in an appropriate way to the T cells? Those are called APCs, for antigen presenting cells, as you can see. So what are some examples of these antigen presenting cells? Dendritic cells, macrophages, and B lymphocytes, okay? Dendritic cells, if you hopefully remember, they're found just under the skin and on your connective tissue, and they basically are your first line of defense. They are the first cells to come in contact with a potential troublesome pathogen or antigen. Then they process it further and present it to the T cells for further action. So it's like a hierarchy, right? So first, the lower rank of soldiers is activated, then the higher rank, and then it goes all the way to the Marines and the Navy SEALs at the very top or something like that. So but for this antigen processing to take place, you need the intervention of something called MHC, and that stands for Major Histocompatibility Complex. What is that Major Histocompatibility Complex? It's a group of proteins which are specific to each individual, okay? Each one of us has a unique type of genetically inherited MHC. So, and there's two types of those MHCs, major histocompatibility complexes, type 1 and type 2, as you see. So MHC type 1 is found on all cells with a nucleus, which is pretty much all body cells with the exception of your red blood cells. Remember, red blood cells do not have a nucleus. And MHC2, which is your major histocompatibility complex type 2, is found in addition to MHC1s, MHC2 is also found on APCs, your antigen presenting cells, which are again these three: dendritic cells, macrophages, and then the B lymphocytes, okay? All right, so MHC1, MHC2, remember that. So let's talk about MHC1s first. So these are glycoproteins, proteins mixed up with some sugar, right? Glycoproteins, type 1. They are genetically determined and unique to each individual. Even identical twins, which basically split from the same egg, have somewhat of a unique and different MHC class 1 molecules, okay? So what do these MHC1 molecules on your immune cells do? Okay, so what they do is uh, they basically get a hold of a potential pathogen or an antigen. If this antigen is recognized as belonging to the body itself, like some of the body cells, then they're ignored, they're friendly, right? However, if fragments from this antigen are recognized as a foreign invader or non-self, that's when the MHC class 1 molecule will latch onto that and present it to the CD8 positive cytotoxic T cells, okay? So that then they can destroy this foreign invader in time. So what you have to remember is MHC class 1 molecules go with CD8 positive cells. So MHC class 1 goes with 8, okay? That's what you have to remember. So MHC1 presents and attaches to CD8 positive cells or the cytotoxic T cells, okay? Cytotoxic T cells. All right, so then, as you can see here in this picture, right, you see the here MHC class 1 molecules, they attach to an antigen. The antigen is those spheres in purple, the purple balls that you see here. MHC class 1 molecule binds to them, processes them, and then presents them to the CD8 positive cells or the cytotoxic T cells, all right? And let's see, where are the cytotoxic T cells here? Class 1 molecule and cytotoxic T cells. All right, they will come up in some other picture later on. All right, so how about MHC class 2 molecules? First up, these MHC class 2 is only found on APCs, antigen presenting cells, which again are dendritic cells, macrophages and B lymphocytes. All right, so those three are the types of cells that carry MHC class 2 molecules. So what do these do? MHC class 2 molecules will actually communicate with the helper T cells or CD4 positive cells. So in other words, MHC2 goes with CD4 positive or helper T cells, okay? So how I remember this is 1 with 8 and 2 with 4. So 4 is like a multiple of 2, so 2 with 4. So MHC2 attaches to CD4 positive cells, and MHC1 attaches to CD8 positive cells. So that's how I remember which is which. So here you can see the MHC2 class molecule in pink, and the antigens are in this green color, the green circles, right? They attach to the MHC2 class molecule. Further processing is done, and then they are presented to your helper T cells or CD4 positive cells for further processing here in this picture. So there you go. MHC class 1 proteins, they attach to the antigen and present it to the CD8 positive cells, which are cytotoxic T cells. MHC class 2 proteins here attach to an antigen and present to CD4 positive cells, which are your helper T cells, all right? So again, 1 with 8, MHC class 1 with CD8, and 2 with 4, MHC class 2 with CD4. All right, so at this point, this also explains why if you get an organ donation from someone who is not your identical twin, right? Because identical twin are very similar in terms of their genetic makeup. So they would be the best candidates for donating their organs to you because your MHC antigens will recognize that organ as not being a foreign tissue. But if it's anyone else with a slightly different MHC proteins on their cells, then you have a problem. Now your immune cells will attack the donated organ and try to destroy it, right? So therefore, the best donors would be an identical twin, then your blood siblings, then your parents, then your first cousins, then aunts, uncles. So you go in that order, right? So that's why before you donate organs, you test a potential recipient of that donation for their blood type and also for their MHC antigens, okay? So you would like ideally to get the blood type from someone with a similar blood type or someone who is a universal donor, but also the MHC antigens, okay? All right, so there was this interesting study that was done at the University of Scotland, I believe, having to do with these MHC antigens. Okay, so let me just quickly make a note here. Okay, University, let's call it University of Scotland uh, T-shirt study. Okay, so what was done here was they took 22 undergraduate male students and they were asked to wear the same T-shirt for 3 days and nights on end without changing. Okay. Then they also recruited 22 young female undergrad students from the university. And what they were required to do was, after the male students had worn that T-shirt for a number of days, T-shirts were taken off. They were packaged in plastic bags, and then these female volunteers for the study were asked to sniff those T-shirts and then um, basically categorize them based on how pleasant they smelled to them. All right. And so basically, what they did was, at the end of the study, they correlated how the females in question correlated the smell uh, from certain male students and how pleasant was that smell to them. And they found something extremely interesting. What they found was that the more different their MHC antigens were of the male student, in other words, the more different the immune system of the male students was from the female student sniffing the t-shirt, the more attractive that t-shirt was deemed to be, okay? But there was certainly an upper limit. If the MHC antigens were way too different, right at the opposite pole, then those shirts, those smells were rejected as well by the females. So you had to be somewhat different from the female's immune system, but not too different. You had to be in that sweet spot in order to be considered attractive, at least odor-wise. Um, and so what that tells us is that nature, at a very subliminal, subconscious level, is trying to pair people of two different genders, sexes together based on slightly differing immune systems. Okay, extremely interesting. So what nature is trying to do is it's trying for us to pair up with people sexually who are slightly different in terms of their immune system, so that the children that are born get a unique type of contribution from both. Okay, you don't want to be marrying someone who's very close to you, but at the same time, someone who's very different in terms of genetic composition is also rejected. Okay, interesting study. And you might be wondering what gave anyone this crazy idea of doing this experiment. Apparently, they had tried it on rats first, and they got similar results. So somebody got the bright idea of replicating the study on humans, okay? And then you might be wondering, well, how did they know about the immune systems of all these people? They took blood samples and then they studied the MHC proteins on the surface of their immune cells, all right? So fascinating study with fascinating results that came out of this, okay? All right, so formation of lymphocytes, both T and B lymphocytes, they are found, actually formed in your red bone marrow, as I mentioned, right? And then they migrate from there to your thymus for T cells. Your T cells get activated in the thymus, okay? Uh, Actually, they mature in the thymus, but they get activated in your secondary lymphatic structures. Now, remember what those were. Those were your lymph nodes, those were your malt tissue, your tonsils. That is where they get exposed to the antigens for the very first time, and then they become activated, okay? And so the effector response is uh, how they launch an effective attack against foreign invaders, against viruses, bacteria, pathogens that might attack the body. So the T cells, they migrate straight to the site of the infection, okay? And the B lymphocytes, they stay back in the secondary lymphatic structures. And what they do is they're kind of like the air force, the air support for the ground troops. Think of them like that, right? So the B lymphocytes will synthesize and then release large quantities of antibodies to neutralize the infection. So they both do their jobs uh, in different ways. And antibodies actually get to the infection site by means of your blood flow and lymph flow, those two, okay? So here in this picture, you can see your red bone marrow is giving rise to all kinds of immune cells, both B and T cells. Then the pre-T lymphocytes migrate to your thymus here, thymus gland. This is a child because you can see that the thymus is quite large at this point. So the T cells mature in the thymus, right? Uh, and even though they have matured, they're still called naive. immunocompetent T lymphocytes. Why are they called naive? Because they have not actually encountered a real life pathogen yet. They're still naive. They're competent. They have been passed and selected and processed by the thymus, but they haven't encountered the enemy just yet. So when they do become non-naive or immunocompetent T lymphocyte, non-naive lymphocytes, once that they migrate from your thymus to your tonsils or your lymph nodes or your secondary lymphatic structures or your spleen and come in contact with actual pathogens and start working, that's when they become mature, active, immunocompetent lymphocytes, okay? So here, these are your secondary lymphatic organs where your T cells become hardened soldiers, where they come in contact with the actual pathogens and mature in your tonsils, spleen, and the lymph nodes, your secondary lymphatic structures, okay? And here are your B cells that are releasing antibodies for air support, if you will, to destroy the pathogens, right? Antibodies are being secreted here as well. All right, so let's try to answer these questions. How is an antigenic determinant related to an antigen? So an antigenic determinant is that part of the antigen, on the structure of the antigen that excites your immune system the most. What distinguishes a hapten from an antigen? A hapten is a micro, small antigen that needs to be uh, uh, connected or attached to a larger protein in order to be recognized as an antigen. Example, poison ivy toxin, pollen, those kinds of things. What features distinguish the receptors of helper T lymphocytes, cytotoxic T lymphocytes, and B lymphocytes? All right, so you have different types of receptors. You have the T cell receptors, the CD4 type receptors for helper T cells, the CD8 type receptors for cytotoxic T cells, and the B cell receptors for the B lymphocytes. Okay. Which type of MHC class molecule is found on all nucleated cells? MHC type 1, and is used to communicate with cytotoxic T lymphocytes. That's MHC1. Which classes are displayed on antigen presenting cells like dendritic cells, macrophages, and B lymphocytes? Both MHC1 and MHC2. APCs have both of them, right? And which class is used specifically to communicate with helper T cells? 2 goes with 4. So MHC2 communicates with CD4 or helper T cells. And cytotoxic T lymphocytes, 1 goes with 8. So MHC1 communicates with CD8, positive cytotoxic T cells. Where does a lymphocyte typically encounter an antigen for the first time? In your secondary lymphatic organs, the tonsils, the lymph nodes, or the spleen. So the secondary lymphatic structure, yes. All right, so let's follow along in the ranks of the T lymphocytes to see how they make their journey to the thymus and how do they pass this extremely rigorous selection process. Let's take a look. So they originate in your red bone marrow. That's where all of them are formed. Then they move to your thymus as pre-T lymphocytes. So these are freshman T cells. Think of them like that, right? It's a fresh core of cadets that have gone for army selection training, okay? So uh, so initially, they are not differentiated yet into helper and cytotoxic types, into CD4 and CD8 positive types. They will be differentiated later on, okay? Uh, and then by the time that they get to the thymus, they have a unique T cell receptor produced randomly based on your genetics. So they each get an ID badge, which is a random ID badge, and this ID badge is generated by your genetically inherited immune system, okay? So now they have to pass 2 types of tests, very rigorous test in this army training ground of your thymus gland, okay? How rigorous is this test? Look at that. Thymic selection eliminates 98% of T cells produced. Almost 98% of your T cells that are hoping to become hardened soldiers to fight pathogens are eliminated. So it's kind of like a Navy SEALs training, very, very rigorous, as you can see here, okay? So what makes it so rigorous? What are the 2 types of trainings, positive selection and negative selection, as you see here. So what is positive selection? So this positive selection looks for those T cells that can actually bind to any other cell in the first place, right? A soldier is no good if the soldier cannot engage another soldier into battle. So they look for cells that are able to bind some thymic epithelial cells within the thymus gland itself. They can engage with them. So those ones are selected. The ones that do not engage the thymic cells, they are weeded out. They're eliminated, okay? So then After being positively selected, then you go on to negative selection. All right, so then what is negative selection? In this part of the selection procedure, you look for T cells that do not bind to your own cells. In other words, you want to prevent soldiers that might turn on their own army. Uh, you want to prevent them from entering the battlefield, right? So that's exactly what negative selection is. So T lymphocytes should not bind to your self-antigens. So those ones are selected. If some of the T cells are binding and reacting to your own body cells, those are eliminated there, all right? So, so thymic dendritic cells, again, present self-antigens, right? It's a test. And the T cells that do not recognize self-antigens as friendly and bind to them, those are destroyed. Those are eliminated, okay? All right, so there you go. This is your selection process, right? For positive selection, uh, you want to see if the cells even bind to any cell. Does it bind to the MHC molecules? If yes, then they can move on to the second level of selection. If they do not bind to the MHC molecules, they are destroyed by apoptosis, all right? So then they proceed, the ones that make it, they proceed to the negative selection part, where what happens is they are presented with the body's own cells, all right? Like dendritic cells present self-antigens to pre-T lymphocytes. Those that are reacting to your own body cells, they are destroyed now. Only those that do not bind with your self-antigens and recognize them as friendly, uh, those are the ones that survive, okay? What would happen if a cell that was not recognizing self-antigen and attacking it was allowed to proceed onwards? Then you will develop an autoimmune disorder. That's what happens in autoimmune disorders when your immune cells fail to recognize self-antigens and they start attacking their own body cells, all right? And what happens if a cell that does not even bind to any cell is allowed to go on? That's a useless. Then you have an immune deficiency. Your immune cell system is not even engaging with any other pathogens. So this is immunodeficiency. If positive selection is aborted or positive selection is bypassed, then you develop an immune deficiency. Let me write it down here. If positive selection of T cells prevented, then you develop an immune deficiency, right? And if the negative selection of T cells prevented, then you develop autoimmune disorders like lupus, like rheumatoid arthritis, autoimmune disorders. All right. So it's a pretty rigorous process, okay? And so look at that, how rigorous. Only 2% of the cells survive the selection process in the thymus, all right? The rest are just weeded out. And of those 2% that make the cut, then half of them are divided into the CD4 helper T cells, and the other half is divided into CD8 cytotoxic T cells. All right. So at this point, they are still naive, which means they have not actually engaged in actual combat. That will happen later on in the secondary lymphatic organs. All right, so where does the maturation of T lymphocytes take place? In the thymus gland. What would happen if a thymocyte that failed the negative selection test was not destroyed and instead entered the blood to circulate? You can tell me that. Autoimmune disorders, right? In general, how does central tolerance differ from peripheral uh, tolerance in T lymphocytes? So what is central tolerance? If you go back here, or peripheral tolerance. So peripheral tolerance is a form of self-tolerance, which means that the CD4 positive cells formed from T cells that bind to self-antigens, they inhibit an immune response, right? So your immune system has some sort of a tolerance to your own body tissue. You don't want an over strong immune system, so strong that it is attacking its own body. That's basically what the point is here, okay? So antigen challenge, where does this happen? The first encounter between antigen and lymphocyte, it takes place in the secondary lymphatic structures, right? Usually in your respiratory tract because you breathe in some pathogen, maybe COVID-19, I hope not. Or in your GI tract because you eat something which has food poisoning bacteria in it, or it can happen in your genitourinary tract if you've had sex with an infected person and are exposed to an STD causing pathogen, okay? So now we are all set for this combat in between your immune system and the pathogen or the antigen. So, all right, so what happens here? Here's the first signal of the impending battle. There's a direct contact with the MHC molecule of your antigen presenting cells. And remember what MHC molecules are present on your APCs, both MHC1 and MHC2. So the antigen presenting cells present the antigen with the MHC class 2 molecules attached to it, okay? And this is happening again in your secondary lymphatic structures, like your spleen or tonsils or lymph nodes, okay? So then this antigen is presented by the MHC class 2 molecule of your APCs to the CD4 positive cell, which is your helper T cell. Now, if this antigen is not recognized as a potential hostile entity, an enemy, then the T cell disengages quickly. No action needed. All is well. Let go. However, if this antigen is not recognized, uh, it could be a foreign invader, then the contact lasts several hours. Now your helper T cell will stay engaged with this antigen for a number of hours, okay? At which point, the second signal will be sent out by your immune cells. What is the second signal here? The helper T cells now start secreting a chemical called interleukin-2. What does interleukin or IL-2 does? It will stimulate even more helper T cells to join the action, okay? So more and more helper T cells are forming. And more and more of these helper T cells, activated helper T cells will produce even more interleukin-2, okay? So now remember, not all of the helper T cells are going to join the battle. You also have the reserve army, the reserve soldiers who are still sitting on the sidelines, waiting to be called into action at a later time. Those ones are called memory helper T lymphocytes, as you can see here. They're available for future encounters. So you also have a backup army here. So here is your first signal, right? The helper T cell, CD4 positive, binds with the MHC class 2 molecules of the APC. And uh, if there is cause for concern, if this antigen is not recognized as being a self-antigen, then your helper T cell starts secreting increasing amounts of interleukin-2. What does the interleukin-2 do? It activates more and more helper T cells, calls them into action, all right? So this is how your helper T cells, CD4 positive cells get activated. Now, how do your CD8 positive cytotoxic T cells get activated? Let's take a look here. The first signal here is a direct contact between the cytotoxic T cell, the CD8 positive cell, and an MHC1 class molecule. Remember, 1 with 8. So MHC1 molecule will interact with CD8 positive cytotoxic cells, okay? And so, and then the second signal is sent out. What is that? The second signal here is again, first interleukin-2, which was released from the CD4 positive helper T cells. So this interleukin-2 would also stimulate more and more cytotoxic CD8 positive cells to join the battle, okay? So interleukin-2 is the big signal here. It's calling all kinds of immune cells here to join the action. But again, even with your cytotoxic CD8 positive cells, not all of them join the battle. Most of them do, but some of them stay back as the reserve army soldiers, the memory cytotoxic T lymphocytes. They stay back. And they will be joining the battle upon re-exposure to the same antigen sometime in the future. This is active immunity, by the way. Once you've been exposed to a virus or a bacterium and you've developed T cells against it, not all of them wipe out the infection during that first battle. Some of them stay back. So sometime in the future, if that same virus or a similar virus comes and tries to attack you, that's when your reserve forces are mobilized at that time. And so they will wipe out the invaders in no time whatsoever. You won't even have an infection. You won't even be aware that you were infected with it. And that is called a subclinical infection. Uh, you know how they keep saying that 80% of cases of COVID-19 have been spread by asymptomatic carriers, carriers who had the virus in the system but were showing no symptoms, but they still got others infected, right? So let's take a quick note here. Asymptomatic COVID-19 carriers, carriers, and um, and memory T cells. Okay. So you might be wondering, well, what is it about these asymptomatic carriers? Why don't they develop symptoms? Why don't they fall sick? Uh, and even though they do carry the virus within themselves and they can make other less genetically fortunate or less immune system fortunate people sick. And the answer is they likely came in contact with either COVID-19 or some related coronavirus sometime in the past, but it was like a minimal dose, and they had those viruses wiped out by their actively working cytotoxic T cells. But at that first time of exposure, they also formed some backup memory cytotoxic T cells. Okay, so when the COVID-19 came about again, those pre-made reserve cytotoxic T lymphocytes attacked and destroyed the infection even before they became aware of there was an infection going on. All right. But they can still shed the virus and make others sick, especially if the other person is over 65 years of age. Because remember, kids younger than 5 years of age and elderly older than 65 years of age, their immune systems are like on the down. They're not as vigorous as they used to be. So those are high risk people. People with diabetes, there's four conditions. 99% of people who have died and gotten seriously sick and ill and required ventilators uh, because of COVID-19 infections were these four categories, right? Uh, and who were those four categories of people? People who had obesity, number one, people who had diabetes, type 2, people who had high blood pressure or hypertension, and people who had congestive heart failure or congestive heart disease. These four people. Almost all the other people, or they were the elderly or very young. Almost all the other people, they do not fall that seriously ill with COVID-19. Okay. All right. So in this picture, we can see how this whole CD8 positive signals thing happens. So here's the first signal. The CD8 positive cytotoxic T cell binds with an MHC class 1 molecule, okay? And then uh, after this, it is stimulated by the interleukin 2, which is released by the activated helper T cell, okay? And so it results in the cloning of this activated cytotoxic CD8 positive cells. See how it's making more and more clones of itself? So they're all gearing up for battle. Okay, so those were the T cells, the CD4 positive helper T cells and the CD8 positive cytotoxic T cells. How about the B cells now, B lymphocytes? How do they get activated? So here's the first signal. So first of all, the antigen binds to the B cell here, right? The B cell first engulfs, it literally eats up the antigen, processes it, and then presents this antigen to the helper T cells for recognition. Again, remember what I said about helper T cells, they are the backbone of your immune system. So everything goes to your helper T cells for further processing, right? So even the stimulated B cells present the antigen because they are antigen presenting cells themselves, the B cells, to the helper T cell for recognition. And then the second signal comes into effect. What is the second signal? Again, the activated helper T CD4 positive cells release something called interleukin 4, not interleukin 2, but interleukin 4, which stimulates B lymphocytes. So that's the difference. Interleukin 2 was stimulating the T cells. Interleukin 4 would be stimulating the B cells. And both interleukins come from your helper T cells. All right, there you go. Here's your B lymphocyte. The B lymphocyte comes in contact with the antigen, and then the interleukin 4 is secreted by your helper T cells, activated helper T cell, and so more and more uh, B lymphocytes are formed here, and more of your B lymphocytes are releasing antibodies now to fight this antigen over here. Okay, so there you go. The plasma cells is what are formed from your B cells, and these plasma cells further produce antibodies. Okay, but not all plasma cells. Some of them stay back. Again, you also have the reserve cores of your memory B lymphocytes, as you can see, that will join the action at a later time should there be a reinfection with that germ. Okay. All right, so what type of cell is required to activate both helper T lymphocytes and naive cytotoxic T lymphocyte? What is the backbone of your immune system? Helper T cells, CD4 positive cells. That's what's needed. How do cytokines released by helper T lymphocytes participate in the activation of both helper and cytotoxic T lymphocytes? Which cytokine are we talking about here? Interleukin 2. Interleukin 2 activates both helper and cytotoxic T cells. Most of them become activated. A few of them become memory cells, means they join the reserve cores and will be active at a later time. Is a separate antigen presenting cell required for B cell activation, or is a B cell able to serve the role of an APC? The B cell itself can also serve the role of an antigen presenting cell. Okay. Explain the role of cytokines that are released from helper T cells in the activation of B lymphocytes. Which type of cytokine activates your B lymphocytes from the helper T cells? Interleukin 4. What advantage is provided by lymphocyte recirculation? More and more T and B cells are formed. It's like enhancing the response, right? Um, Amping up the response to the infection. All right, so what happens here is your activated helper T cells. See, it's releasing more and more of the interleukin 2, the cytokines. And this reminds me of the cytokine storm as well. Remember we talked about the cytokine storm? A good number of COVID-19 sufferers actually uh, fall seriously sick or even die because of what is called a cytokine storm. What does that mean? It's not actually the virus itself that's killing them, it's their own immune response, exaggerated, overstrong immune response that causes tissue damage that results in their complications and death at times. And that's what a cytokine storm is about. So many cytokines like interleukin 2 are being secreted that their immune system goes into an overdrive, all right, way more exaggerated response, kind of like an autoimmune response, right? And so it destroys their own kidneys and uh, heart and brain and their own body tissues. Most of these deaths happen because of either respiratory failure, because the lungs basically fill up with fluid, or kidney failure. Okay. Again, this is all autoimmune. All right. So in this picture, we are seeing how your activated cytotoxic T cells or CD8 positive cells destroy the pathogen. They destroy it by releasing two types of chemicals, perforins and granzymes. Perforins, even the name tells you what it does. It perforates, punctures holes in the cell wall of the attacking antigen and destroys it. Granzyme is kind of like a destruction kit, right? These are proteins, granzymes, that will shrivel up the offending virus or bacterium. That's something called apoptosis or programmed cell death. That's apoptosis, okay? So perforins and granzymes, two ways um, that your cytotoxic T cells can destroy antigens. Okay, are cells of both the innate and adaptive immune systems regulated by cytokines released by helper T lymphocytes? Absolutely, yes. Cell-mediated immunity is specifically effective against what type of target? Against viruses and tumors, okay? What is the specific role of plasma cells? Plasma cells release antibodies. So antibodies is what we're talking about next, also called immunoglobulins. What does that tell you? These are parts of your immune system, and they're made up of proteins called globulins. Where do you have globulins? In your egg white. Remember we talked about how eggs are superfood because they have the three fundamental proteins in them. They have albumin, which gives shape to your cells and the tonic power to your cells. Then you have the um, globulins, which make your immune system, literally your antibodies, and then fibrinogen, which is found in egg yolk. And fibrinogen forms fibrin, which makes your blood clot. Okay, so all these uh, critical proteins you can get from eating eggs. Okay, so antibodies, what are they? Antibodies are structures like these. They look like the letter Y. That's what an antibody looks like, okay? And so here you have what is called a light chain of the arm, and these are the heavy chains of the arm, right? And then these are the antigen binding sites. So these are the places on your antibody where an antigen or a pathogen will attach here. And these antigen binding sites are of different shapes. So, and that's what specific immunity or adaptive immunity is all about. For each virus, this antigen binding site would be a different shape. Not all viruses look the same. The influenza virus looks very different from the coronavirus, and the coronavirus looks very different from HIV, the human immunodeficiency virus, which yet looks very different from chickenpox virus or something, right? So based on each viral shape, this antigen binding site of your antibodies would be different. It adapts. It adapts according to the shape. That's why it's called adaptive immunity. How can you keep your adaptive immunity in perfect form? Eat healthy, exercise, outdoors, maintain your stress levels, and get adequate sleep, and hydrate yourselves. Okay, these are the things we have to follow. Okay, next up, we are looking at some ways that your antibodies can destroy antigens and pathogens. Okay, the first way they can do this is by something called neutralization. What happens here? Look at that. Antibody neutralization, antibody literally covers the biologically active portion of a microbe or toxin. So here's the virus, looks like the influenza virus. Influenza virus almost looks like those dart balls that you throw at the target and they stick. Uh, that's pretty much what it looks like. Those spikes, they're used for attaching to your throat cells to make you sick. So look what the antibodies are doing. They're attaching to the very active sites, those attaching sites of the virus, so the influenza virus cannot attach. It cannot latch onto your cells, cannot make you sick. So that's called neutralization. Something else that the antibodies can do is they can play this game of like ring around the rosies, kind of, you see how they're kind of holding hands and they're just uh, holding all of these bacteria prisoners. They're holding them captive. This is called agglutination. Antibodies cross-link cells, for example, bacteria forming a clump. So they deactivate and take out the bacteria out of the action by doing this. This is called agglutination. Literally means clumping. Or they can do precipitation. What is precipitation? Where the antibody cross-links circulating particles, forming an insoluble antigen antibody complex. They can simply attach to the offending pathogens, the antigens here, and so this neutralizes the whole thing. Now, this whole uh, complex that you see here, the antigen antibody complex, will be devoured by a macrophage, right? A large Pac-Man cell would come and eat up the whole stuff, thereby neutralizing the virus. All right, three other ways that your antibodies can fight off pathogens. Let's see. So here, the first thing is called complement fixation, okay? What is that? So complement fixation, what happens here, here's the antibody, the Y-shaped thing, right? And see how here's the active binding site of the antibody, which is attaching to the bacterium. It attaches the bacteria on the one end, and the other end of the antibody is attached to a protein called a complement protein, okay? Complement. And the complement proteins are secreted by your liver, and this complement protein will actually destroy the pathogen here. So it's like bringing the robber or the criminal to the police, to the cop here. Here's the cop, here's the robber, and here's the neighborhood watch guy who got hold of the cop and is bringing him to the, who got hold of the criminal rather, and is bringing him to the cop here. So that's the situation. That's complement fixation. Then you have something called opsonization. What does that mean? Look at that. The two antibodies attach to the bacterium and pull it all the way into the jaws, the waiting, eager, hungry jaws of a macrophage or a phagocyte, which is going to eat up the bacterium. That's called opsonization. Or finally, what the antibodies can do is they can activate uh, what are called natural killer cells. And they are pretty much like macrophages. The natural killer cells will come and engulf and eat up the whole thing here as well. So these are the 6 ways that uh, antibodies can basically neutralize, get rid of these viruses, okay? So speaking of immunoglobulins or antibodies, there are 5 of them, 5 classes: IgA, IgG, IgM, IgD, and IgE. Let's take a look at each. By far, the most common type of antibody in your system is the IgG type. Look at that, 75 to 85% of your antibodies in your blood are IgG type. These are also the types of antibodies that get activated in a secondary infection, when the infection happens a second time. IgG are also the only type of antibodies that can pass across the placenta from the mother to the unborn child. So therefore, um, remember how we talked about Rh incompatibility between mother and child? If the mother is Rh negative blood type, the child is Rh positive blood type. Um, no problems during the first pregnancy and childbirth, but if the second child that is being, or the fetus that is being conceived is again Rh positive, then the mother's preformed antibodies can pass across the placenta and destroy her own child. Those would be the IgG type of antibodies. They are the only type that move across the placenta. Okay, then you have the IgM type of antibodies, and these antibodies are critical during your primary response. The first time that you get sick with a virus or a bacterium, the first antibodies that rise in numbers would be the IgM type. All right. Okay, IgA, how about these antibodies? These ones are important because they are found in your body secretions. What are your body secretions? Sweat, saliva, tears, mucus, breast milk, semen, vaginal fluids, uh, prostatic fluids. All of these fluids that you expressed to the outside of the body, your exocrine glands, they contain IgA. And that is why breastfeeding is so much encouraged to all mothers, right? Because when you breastfeed a child, what you're giving the child, along with all the excellent nutrition in your breast milk, is these types of antibodies, IgA. And guess what? The mother is passing on the IgA, the antibodies, which are tailor-made against the very pathogens that she was exposed to during her lifetime. So what better gift that a mom can give to her child? This immunity at birth by breastfeeding the child. All right, so that's why breastfeeding is so encouraged, that you pass on the IgA type of antibody to the child as well. And then finally, you have no, not finally, you have two more. So IgD type antibody is basically, it functions as an antigen specific B lymphocyte receptor. IgE antibodies, these ones are important because these are released in allergic reactions, right? So if you are allergic to wasp venom, as an example, or fire ant stings or something like that, why that happens is the first time that you were bitten, your T cells reacted to the venom, and so did your B cells, and they got sensitized to that type of venom. The second time that you're stung or bit by the same creature and you are exposed to the same venom, now your plasma cells start releasing IgE types of antibodies by the millions. And these IgE antibodies cause the symptoms of an allergic reaction, the hives, the rash, the itching, the breathing problems, all of that, right? They happen because of the IgE. So that's what you have to remember. IgE type of antibodies are related to allergic reactions. All right, so what is the significance of the variable regions of an immunoglobulin molecule? The variable region basically changes according to the shape of the virus. That's the adaptive part of the immune system, of your antibodies. What are the six major functions of antibodies? Six major functions: opsonization, neutralization, complement fixation, natural killer cells, immune complexes, and precipitation. Uh, which occur due to antigen binding, complement fixation, and also what else? Opsonization. And which depend on the FC region, the complement fixation, depend on the FC region. Which subclass of antibodies is most prevalent? IgG type. Which specific functions of antibodies can it engage in? IgG can do all kinds of things. It can cause neutralization, opsonization, complement fixation, all of those. They're also the only type of antibodies that can pass across the placenta from the mother to the unborn child. Okay, so what is immunologic memory? Immunologic memory is the simple fact that some of your T cells and B cells were acting as memory cells. They did not join the battle right away, but they were sitting on the sidelines as reserve players, waiting for a secondary infection to come along so that they can engage it again and destroy it. That's what immunologic memory is all about, all right? So that's a very powerful secondary response. These are your, again, reserve core reserve armies corps that already know how to tackle an infection, and they're way more effective and swift and lethal uh, during their secondary response as compared to a primary response. So that's why a person who develops measles will not have it again, even if exposed, because they have this powerful secondary response still hanging around from the first time that they got exposed to it. And this is also the reason why somebody who has chickenpox once doesn't get chickenpox again. They might get shingles, which is a very localized, mild form of chickenpox, but they won't develop the full-blown infection again. All right. So here, like, to take a look at this graph, look at the difference. So here's the primary response. The first time that you were exposed to a pathogen, what was the first class of antibodies that came into action? IgM. But that was not much of a response. It took about 10 to 15 days and then just kind of just tapered off. The IgG type of antibodies started rising much later. They were a little bit more effective, but then they tapered off as well during your primary response. What happened during your secondary response should you be re-exposed to the same virus again? Now look at that. From the get-go, from as soon as you're infected, from day zero, look at your IgG antibody. They're going crazy all over the place. Look at how many IgGs are being generated. They're going to destroy the infection ever before you even become conscious of the fact that you were infected with it. And there's a much, much weaker IgM response to it. It doesn't matter at that point. Okay, so why is there this lag time between the uh, between the time that you're exposed to a virus and the time that you develop antibodies to it? We all know that we fall sick with the influenza virus, flu virus, or the cold virus, and it takes us about like a week to 2 weeks to recover. So why is this lag time there? Because our immune system is still getting used to the new virus or bacterium that has attacked our system. It's still getting immunized. It's still getting used to it. So it takes the antibody levels to build up to those effective levels about 5 to 10 days. So that's what this lag or latent phase that's all about. All right, so here, two types of immunity that we're looking at, active versus passive immunity. What's the difference? Simple difference. In active immunity, your own body cells, immune cells, make their own antibodies, all right? So active immunity is conferred when your own B lymphocytes have to make their own antibodies. So how do you get them to do that? Two ways. Either you get sick with the disease and your B lymphocytes come in contact with that pathogen and then release antibodies against it, right? And you recover to become immune to that. That's one type of active immunity. The other type of active immunity is you get a vaccine. When you get a vaccine shot, what that does is the vaccine puts in a weak or a dead virus into your system so that your B lymphocytes can recognize that virus and launch an antibody attack against it, so your levels build up. So then the next time around, should the real infection come inside of your system to attack you and infect you, you already have those preformed antibodies from your vaccine. So active immunity forces your B lymphocytes to make their own immune cells, make their own antibodies, right? And there's two ways of getting active immunity: either get sick and then recover, or get a vaccine. So what is passive immunity? Passive immunity is a gift of antibodies from some other source. You are not, your cells, your B lymphocytes are not making their own antibodies. Rather, you're getting this gift of antibodies from some other source, okay? So passive immunity is again of two types. One is passive uh, natural immunity, which is what? Breast milk. Your mother breastfed you and passed on her preformed IgA type antibodies to you. Now you have passive immunity. All right, you got this gift of preformed antibodies from your mom. That's natural, passive natural immunity. The other type of passive immunity is something called passive acquired immunity, uh, something also called inoculation, okay? Which means what? That you receive a gift of preformed antibodies by means of an injection, inoculation, all right? Inoculation is passive immunity. Let me write it down here. Inoculations and breastfeeding are types of passive immunity, while what? Falling sick and recovering from an infection, recovering from infection, as well as uh, vaccination. These are types of active immunity. Okay, these are active immunity. Okay. For example, you're bit by a rabid animal. Okay, and remember, in America, the single animal that is responsible for most cases of rabies, it's not dogs or skunks or coyotes or raccoons even, it's bats actually, all right? Bats cause most rabies cases in the United States, all right? So again, quite common among cave explorers, people who go to caves, study bats and stuff like that, also areas where bats are common, okay? And so the virus that causes rabies, the rabies virus is one of the most fatal viruses known to man. After the virus has made its way through your nerves to your central nervous system, at that point, the fatality rate, the death rate is almost 100%. Okay, so you have a very narrow window to act. So if you are bit by or exposed to the rabies virus by whatever means, and if you're transported to the ER right away, they can give you uh, antibodies against the rabies virus. They can inoculate you. And these antibodies are collected from other people who were immune. Now they can also synthetically manufacture rabies antibodies and give you that shot. Okay, so that would be that's called an inoculation. So the difference between an inoculation and a vaccination is what? A vaccine forces your immune cells to make their own antibodies. That's active immunity. And inoculation is simply a gift of preformed antibodies infused into you from a source. Okay. Now remember, the only way that you can form long-lasting immunity or immunologic memory is with active immunity. Passive immunity, when you get a gift of preformed antibodies, you don't form any memory cells. So it's very short lasting. It'll save your life for that period of time, but then those antibodies go away. Only with active immunity can you develop a immunologic memory, all right, long-term memory. So here, this is a good table. Active and passive active immunity is production of memory cells due to contact with an antigen, naturally acquired active immunity, like direct exposure to the antigen, and then artificially acquired from a vaccine. Passive immunity, no production of memory cells, right? Naturally acquired, like from mother to child through breast milk, and artificially acquired when you get an inoculation, preformed antibodies from some other source. So these are what vaccines are. They are basically shots of dead or weakened virus that are given to you so you can develop your own immune response to that virus. So should it come back at a later time, the real thing, now you have preformed memory cells against it, okay? It may give you lifelong immunity, or some of them require a booster shot. All right, you might need boosters as well. Like for tetanus shots, you need boosters every 10 to 15 months. All right, so hypersensitivity, what is that? Hypersensitivity would be, all right, the hypersensitivities are exactly what it says, right? How your immune system reacts in an exaggerated manner, hypersensitive to certain normal antigens, so-called normal antigens, right? Like people who might have pollen allergies. Not everyone is allergic to pollen, right? So those who do have a pollen allergy have a type of hypersensitivity. So what are the different types of these hypersensitivities? The first one is the fast occurring ones, acute hypersensitivities, like people who are allergic to wasp venom or bee stings or those kinds of things. It happens fast. That's an acute hypersensitivity. It could be life-threatening as well. Something called an anaphylactic reaction, right? Anaphylaxis. So these people have to carry those EpiPen shots with them, epinephrine. So every time they become exposed to the trigger, they need to take that shot. And what adrenaline or EpiPen shot does is it opens up their breathing passageways, also strengthens the heart, and prevents their blood and plasma from leaking out of the blood fluids, because most of people die or face complications because of circulatory failure, all right, or breathing respiratory failure. So that's what you're trying to prevent. Subacute hypersensitivity take anywhere from 1 to 3 hours to develop, and both acute and subacute hypersensitivity sensitivities are caused by IgE type of antibody. So then you have delayed hypersensitivities that take from a couple of days to manifest themselves, and these ones involve your T cells. All right. Okay. So acute hypersensitivity, as I mentioned before, like against pollen, peanuts, wasp stings, those kinds of things. And we all know the signs and symptoms of sneezing, runny nose and watery eyes, a throat that seems to be closing in on itself, vomiting and diarrhea. In serious cases, it might cause severe inflammation and what is called an anaphylactic shock, circulatory failure. So now we're looking at immune deficiency, and the one of the most well-known infectious agents for that is AIDS, which stands for acquired immunodeficiency syndrome. This disease is caused by the infection with HIV, which is the human immunodeficiency virus. And what this virus does is it destroys your helper T cells. And remember how the helper T cells, the CD4 positive ones, are the very backbone of your immune system. So when you destroy them, everything else falls apart. So these people literally have no immunity against the so-called common infections, okay? And how can you get AIDS, or rather uh, be exposed to HIV? There's four ways. One is by sharing needles, okay? So for like drug abusers, people who share needles, or uh, you know, uh, any instrument that requires blood swapping. Sexual transmission, okay? And it used to be that it was more common amongst people like gay men, okay? Now, most of the transmission takes place during heterosexual contact from the male to the female partner, okay, mostly. Then the third way you can get it is by unscreened blood transfusions, which is fortunately quite rare in the developed world. And then finally, from the mother to the unborn child, which is called vertical transmission, okay? So sexual contact is a lateral transmission, sideways, and vertical transmission is from mother to child, down. So how do you know what's the difference between being HIV positive and actually having AIDS? HIV positive simply means that you have the virus in your system, and you can detect that virus by means of different studies uh, like the ELISA test, right? ELISA test for ELISA, that stands for enzyme linked uh, immunosorbent assay. In other words, you are just simply looking for antibodies against HIV. If you have high levels of that antibody, well, you have been exposed to HIV, right? Um, but simply being HIV positive doesn't mean you have AIDS. AIDS is declared as the diagnosis when a person's white blood cell count falls to less than 200 cells per cubic milliliter. In a normal person, they should be 12,000 to 20,000 immune cells per cubic milliliter. But someone with full-blown AIDS, that number falls below 200 cells per cubic milliliters. When you get to that low level of immunity, even a common cold will prove fatal for you. So death is usually from opportunistic infections or cancers, right? So many infections such as what is called a PCP infection, PCP, that stands for Pneumocystis carinii, Pneumocystis uh, carinii pneumonia. It's quite rare. It almost always is seen in people with immune deficiency, like with AIDS sufferers, okay? Or another infection called Kaposi sarcoma. This is a rare tumor, which is almost always associated with immune deficiency as well, right? Another one is Burkitt's lymphoma. Again, quite common in Burkitt's lymphoma with AIDS. So when these kinds of opportunistic and rare infections start arising in a person, you know that the immune system is pretty much gone at that point. So prevention, again, safe sex, use protection, condoms, diaphragms, right? Abstinence, all of those. Screen your blood products, don't share needles. And if you're a mother, get treated because you can pass this on to your child too. So what is immunologic memory? Your T cells forming and B cells forming reserve cells that can join the battle at a later time. How does the secondary response differ from the primary response? Secondary response is much swifter and more powerful. What is the advantage of the secondary response? Very swift. You develop subclinical infection. You do not even get sick. Which type of immunity, active or passive, results in the production of memory cells, active immunity, and generally provides lifelong protection from that antigen. All right. So then let's go ahead and start our discussion of the next chapter, which is the respiratory system. And it has two parts. Today, hopefully, we will go over the the first part here. So what is your respiratory system? Of course, your breathing apparatus, right? Everything from your nose back to your pharynx, then larynx, down to the trachea, then the bronchi and the lungs, and to the very minuscule levels of the lungs as well. So that is your respiratory system, and you need it for exchange of gases, take in oxygen, give out carbon dioxide, right? It also helps you detect odors. You smell through your respiratory system. You also speak and make sound, making use of your respiratory parts of your respiratory system as well. All right, so what you're seeing here is something quite important, the distinction and the demarcation between the upper respiratory tract and the lower respiratory tract, okay? So what is considered upper versus lower respiratory tract? The demarcation line would be your larynx. Everything above the larynx, that includes your nose, paranasal sinuses, pharynx, is considered an upper respiratory tract organ. Everything, including the larynx and lower down, that includes your larynx, then your trachea, then your left and right bronchi, and the lungs, would be your lower respiratory tract, okay? So the upper, so this is your structural difference, as you can see. In terms of function, your upper respiratory tract, um, so the entire area from your nose to your paranasal sinuses, to your pharynx, to your larynx, to your trachea, to your uh, bronchi, down to your bronchioles, down to your terminal bronchioles is called the conducting zone, which means all it does is it carries oxygen, air. No exchange of gases actually takes place. Only when you get down to the respiratory bronchioles and the alveoli, these grape-like structures which are microscopic in your lungs, this is what is called the respiratory zone, because only in this region do you exchange gases. So conducting zone all the way down to your respiratory bronchioles, so respiratory bronchioles down to the alveoli is your respiratory zone, okay? All right, so what is your respiratory system basically made up of tissue-wise? Mucous membranes, and they secrete mucus. That's why they're called that. And anyone who has seen mucus in their noses know what that is, right? So one thing about your respiratory epithelium is it becomes thinner and thinner as you go deeper down, right? So once you are in the nose and the paranasal sinuses, the type of epithelium you have there is pseudostratified ciliated columnar epithelium, okay? Ciliated means it has those little hair-like structures called cilia, which makes sense. Those cilia filter out all the dust and debris and pathogens that you might be breathing in, right? It's a thicker layer. By the time you get down to your lower respiratory tract, like your bronchi and into your bronchioles, it turns into simple ciliated columnar. It becomes thinner, but it's still cilia are there. By the time you get down to your terminal bronchioles, now the type of epithelium is simple cuboidal. And by the time you get down to the very smallest of your lung regions called the alveoli, where actual exchange of gases takes place, then this epithelium changes into simple squamous epithelium, okay? All right. However, if you look at the back of your throat, the pharynx and the vocal cords region, the type of epithelium here is stratified squamous epithelium, because this type of epithelium is the best for providing wear and tear protection, okay? Here, so you can look at the typical epithelia found in your respiratory system, right? So here is your ciliated columnar epithelium. You can see the hair-like cilia, right? All over there, those are the cilia. You have the mucus-producing goblet cells that produce a lot of mucus to trap the dust and the debris here. These are the columnar cells. Here's the basement membrane, and then the lamina propria, the connective tissue underlying it, which is rich with blood vessels. So here, as you go down in your respiratory tract, your epithelium becomes thinner and thinner. So starts out with pseudostratified ciliated columnar epithelium like this, all the way down to your lobar bronchi. By the time you get to the segmental bronchi and the larger bronchioles, it becomes simple ciliated columnar, thinner. Then by the time you get to the terminal and respiratory bronchioles, it's simple ciliated cuboidal epithelium. Once you get to the alveoli, to the very thinnest of your and the most intimate of your lung structures, you have simple squamous epithelium here, okay? So we all produce mucus, and how much mucus we produce depends on who we are, our genetics. Some people produce more mucus than others. Also, where we live, if you live in a heavily polluted area, you generally produce more mucus. If you're a smoker or inhale polluted air, in other words, you produce more mucus than someone who does not, right? What does mucus do? It traps dust, dirt, pollen. That's what it does, right? Uh, and the mucus is quite protective. Look at that. Mucus contains things like lysozyme and defensins and IgA. These are disease-fighting chemicals which are found within. So this is our own inborn body-made defense against inhaling pathogens, germs, and mucus and uh, dust and allergens and those kinds of things. Okay. So who produces more mucus again? People who smoke, people who have exposure um, to polluted environments, people who have allergies, right? People who are suffering from a cold infection, they produce more mucus. So mucus is called sputum when it's coughed up with mixed with saliva and trapped substances. And sputum is quite important in pathological terms because um, you would like to take samples of sputum from someone who has a chronic cough, and you can use this sputum to try to see if there's an infection, a lung infection going on, or even lung cancer, right? So the two studies that you can do on sputum samples are a sputum culture, right? A sputum culture where you try to grow certain bacteria on that sputum that you've collected from a patient. So sputum culture to look for uh, infections, right? Or you can do sputum cytology, where you look at the cells contained within your sputum to look for cancers, to look for respiratory tract cancers, respiratory tract cancers. So those two important lab tests can be done on a sample of sputum. So which respiratory structure is associated with the exchange of respiratory gases? Your respiratory bronchioles and alveoli. What three structures compose the upper respiratory tract? Your nasal passages, paranasal sinuses, and the pharynx. Um, what three structures compose the respiratory zone? The respiratory bronchiole, the alveolar duct, and the alveoli themselves. In what ways does the epithelium of the upper respiratory tract differ from the epithelium in the alveoli? The epithelium of the upper respiratory tract is pseudostratified columnar ciliated epithelium. The epithelium in the alveoli is simple squamous epithelium. So let's start with the nose, right? So your nose is made up of a lot of cartilage, right? There's some bone. The nasal bone is right at the bridge of the nose where you wear glasses, as an example, but the rest of it is cartilage, as you can see here. Okay, so there's the nasal bone right at the bridge of the nose, right? Uh, and this is cartilage. It's called the septal nasal cartilage. And then this is the lateral, the sideways cartilage. And this is something called the alar cartilage. This is the cartilage that you can, when you flare your nostrils, you can actually move that alar cartilage here, okay? And these are some of the inner structures. So this area in blue is your nasal cavity, okay? These are your turbinate bones, the inferior turbinate, the middle turbinate, and the superior turbinate bone. And what these bones do, they cause the air that you breathe in to be warmed, to be moistened, and to be cleansed. Cleansing, warming, and humidifying, three functions that are done in the nasal region. Then this orange part here, that's your pharynx, all right, the nasopharynx. And then the purple, oropharynx. Oral, this is your mouth and the tongue, so this is the oropharynx. And then everything below that in green and downwards is a laryngopharynx. All right, so here is the distinction. All right, so let's take a look at the inner, deeper regions of the nose again here in this picture, right? All right, so what do we have? This part right at the top of the nose, right here. Uh, this is your olfactory region. Olfactory region meaning you breathe, you smell. So your smelling epithelium is found here, the olfactory region. The blue area is your respiratory region, so the turbinate bones, right? Or the conchae, the nasal conchae, same thing here. The green region here is called your vestibule. This is just the entrance to the nose, okay? So the choanae are your nostrils. So your nostrils open in the back of the throat as well. Here's your nostrils, they open in the back of the nose. Those are called the choanae here. What you're seeing here is your uvula as well. The uvula is this fold of connective tissue that hangs at the back of your mouth, right? When you scream, or you must have seen that, the curtain hanging at the back, that's called the uvula. And the function of the uvula is to prevent food from going back up into your nasal cavity when you swallow. That's what the uvula does, okay? So here, this is an actual cadaver, and you can see the different parts of the nose, right? The bones, the nasal conchae, the superior concha, the middle concha, and then the inferior. And what they're also called turbinate bones because they cause turbulence in the air that you breathe in. And the function again is warming, cleansing, and humidifying. Okay, it happens here. You can also see your paranasal sinuses here. Sinuses are these hollow air-filled cavities around the nose. So there's your frontal sinuses, a pair of frontal sinuses. The largest ones are your maxillary sinuses. Those are the biggest ones. You also have a smaller ethmoidal sinus here, right? And a smaller sphenoidal sinus here. So those are the four sinuses that you have, four paranasal sinuses that you have. Okay, so then you have something called the nasolacrimal duct, and it's called that because, let's see here, there's your there's your uh, nasolacrimal duct. It connects the medial angle of your eyes with your nose, and that's why when you cry, you've been crying for a while, those tears actually flow out of your nose, right? Because of the nasolacrimal duct. That's what it does. And what the nose itself does, again, warming, cleansing, and humidifying. That's what nose does. Uh, so that's why when you get like a cold or the sniffles or like a sinus headache because it's too cold, you need to do these three things. You need to warm the air that you're breathing, you need to cleanse it, and you need to humidify that. So when you wear a mask, such as the custom these days, you are doing exactly those three things. You're warming the air that you're breathing in, you're cleansing it, you're filtering out the pathogens and dust, and you're also humidifying it because when you breathe in and out into a mask, it holds a lot of moisture. Okay, especially if you wear a glass, you'll feel your glass fogging up with that. So wearing a mask will help your nose do all of these three functions. So I know the mask mandate has been lifted lately, but wearing a mask, especially during cold, dry season, has its benefits, all right? Rhinorrhea. What that means is a runny nose. And what might give you a runny nose again is allergies, asthma, uh, if you have a cold infection, eating foods that are hot and spicy, even certain emotional states will make your nose run, and exposure to cold air. All right, all of those cause rhinorrhea or a runny nose. Okay, so then we have the paranasal sinuses, and let's take a look at what those look like. Okay, so here they are. So you have those paired frontal sinuses right there at the top. The largest of all are the maxillary sinuses here. Then you have the ethmoidal sinus in green and the sphenoidal sinus in yellow. These are the four sinuses that you have, all right? So these sinuses have the same type of epithelium as your upper respiratory tract, pseudostratified ciliated columnar epithelium, and they also are lined by a mucous membrane, and that mucous membrane releases mucus which is swept to the back of your throat, and you might swallow it or spit it out depending on your preference at that time. All right, so one thing to remember here, the maxillary sinus, this region is also called the danger area or the danger triangle of the face. So the danger area slash triangle of the face. Okay, and why is it called that is, let's say you have a lot of like, uh, you have a chronic sinusitis condition where your sinuses are clogging up and getting filled with mucus and getting infected over and over again, either because you have chronic allergies or you fall sick with a virus over and over again, or if you are simply a smoker, someone who's sensitive to the pollen in the air around you, whatever the reason. uh, when your sinuses get infected repeatedly, sometimes that infection, if untreated, can basically climb all the way up to your brain. Okay, or maybe let's say you're getting a nose job done for a cosmetic procedure, a deviated nasal septum. You're trying to get that your nose is slightly crooked, which is the case in most people, and you want it to be cosmetically fixed. And the surgeon or the ENT surgeon is doing some instrumentation in this region. He or she, if they're not careful enough, they can introduce this infected mucus into certain veins that connect directly to your brain. So the infection deep within your maxillary sinus can actually ascend all the way up to your brain by means of those veins here, venous sinuses. And if that happens, then your chronic sinusitis can become acute encephalitis, an acute brain infection, which could be deadly. And that is the reason why the maxillary area is called the danger triangle of the face. So you have to be careful whenever there's any instrumentation being done there. All right. Sinus headaches, most of us have suffered from that. Why does that happen? Because either due to cold, dry air or allergies or an infection, the mucous membranes in your sinuses become inflamed. So that clogs up the mucus here. And the mucus puts a lot of pressure on the surrounding paranasal sinuses, which are richly supplied with sensory nerves, so that you feel as a throbbing headache. So how do you get rid of it? Uh, Inhale steam. Steam will liquefy your mucus, and so blow your nose gently out, liquefy the mucus, allow it to escape, and that should reduce the inflammation and bring down the sinus headaches. Okay. So here you're looking at the pharynx, which is the back of the throat. Okay. It's divided into three regions: the nasopharynx, the oropharynx, and the laryngopharynx. All right. So the nasopharynx is where you have the back of your nose meeting up with the back of the throat. The type of epithelium in this region is, again, pseudostratified ciliated columnar epithelium. Okay. And the important thing that happens here is whenever you swallow, your soft palate elevates, your uvula, the curtain of tissue at the back of your throat, it rises and prevents food from going up from the back of the throat up into your nose. You also have a very important structure here called the auditory tube or the eustachian tube. The eustachian tube connects your back of your throat with your middle ear. So whenever you travel on an airplane, or there's like pressure changes, you know how your ears pop? That's because of pressure inequality uh, across the eustachian tube. The eustachian tube gets crushed or opened based on the temperature, the pressure changes that are happening. Um, Eustachian tube is also important for another reason, like in children. You know how the middle ear infections are so common, something called otitis media, and that is because they have a much shorter eustachian tube. So any throat infection where they have mucus in their throat or the nose can easily backtrack through the eustachian tube and end up infecting their middle ear. So how do you treat middle ear infections? You put a tube trying to keep the eustachian tube open so the mucus can constantly drain back to the back of the throat. You have to put in a tube there. You can also try antibiotics, okay? And then of course we have the tonsils in this region called the pharyngeal tonsils as well. Okay, then the oropharynx, the epithelium lining your oropharynx is non-keratinized stratified squamous epithelium. You have two types of tonsils in this region, the palatine tonsils and the lingual tonsils. Finally, down to the laryngopharynx, where you have, again, non-keratinized stratified squamous type of epithelium, all right? So here you can see the nasopharynx. Here's the nose connecting to the pharynx. There's your adenoids or tonsils at the back. Here's the eustachian tube connecting the back of the throat with the ear. In kids, infections can easily go in either direction, right? Here's your hard palate. There's your uvula. Here's the tongue. Here's the oropharynx. Here's an extremely important structure called the epiglottis. Epiglottis is a flap of cartilage. Every time you swallow your food, this cartilage, the epiglottis, falls down like a lid to a trap door, and it will close the opening to your trachea. Here's your windpipe. So when you swallow food, this so-called trap door, the epiglottis will fall downwards and close the opening to your trachea, to your windpipe, so the food does not end up choking you. It does not end up going here into your trachea. Rather, it travels back into the esophagus where it should, and then the esophagus carries it down into your stomach, okay? And there have been many cases, especially in people who were uh, heavily drunk or sedated or under the influence of drugs. When you have any of those conditions, of course, your reflexes are depressed, including your swallow reflex. So when these people were tried to be fed or were eating and drunk heavily at the same time, their epiglottis didn't work properly. So the epiglottis didn't shut all the way down, didn't close off the trachea. They ended up uh, swallowing their food into their own windpipe, into their trachea, and that causes aspiration pneumonia. The food ended up in their lungs, causing fatal pneumonia in many cases. Aspiration pneumonia. And if you're a nurse in the ICU with patients who are immobile, lying on their bed with like all kinds of tubes sticking into them, you have to be careful that you elevate the head of the patient at a 45-degree angle. So the stuff, the food stuff, all of their secretions do not dribble down their trachea because they do not have much of a swallow reflex. All right, aspiration pneumonia in heavily sedated slash drugged slash uh, comatose patients. Comatose patients. So you have to be careful as a nurse with that kind of stuff. All right. Okay. So what changes occur to inhaled air as it passes through the nasal cavity? Warming, cleansing, humidifying. What is the function of nasal conchae? Turbinate bones. They cause the air that you breathe in to become turbulent. Again, warming, humidifying, and cleansing. How are the paranasal sinuses connected to the nasal cavity? By means of tubes, paranasal passages. What two regions of the pharynx contain tonsils? The oropharynx, which contains the palatine and the lingual tonsils, and the nasopharynx that carries the pharyngeal tonsils. What is their purpose? Tonsils are your immune organs, secondary lymphatic structures. Okay, so let's take a look at your at the anatomy of the respiratory system here, okay? So let's start with your larynx, also called your voice box, because that's where you have your vocal cords and your vocal cords vibrate to make sound, right? And so you you produce sound there in the larynx, and you also have the epiglottis here, which, as I just explained, prevents food from going down the wrong tube into your breathing tube at that point. So you can also produce something called the Valsalva maneuver. What is the Valsalva maneuver? Such as when you're straining on the pot, when you're trying to defecate, you know how you, uh, or when you're throwing up, when you vomit, when you're giving childbirth, when you're giving childbirth and you're kind of contracting your abdominal muscles, that is the Valsalva maneuver, all right? So you contract your abdominal muscles and you close the opening to your larynx at the same time. That increases your intra-abdominal pressure, which will help with urination, defecation, and childbirth. That's the Valsalva maneuver. But don't do it too often because that can cause hemorrhoids because of and also hernia, where your intestines actually pop out of the covering. And also when you do the sneeze and cough reflex, that's when the Valsalva maneuver also kind of happens, okay? All right, so here is your larynx. Right at the top, here's the trachea, here's the larynx. So the front of the larynx is uh, protected by the thyroid cartilage. This is the thyroid cartilage, also known as your Adam's apple. The Adam's apple is the thyroid cartilage right here. Just below the thyroid cartilage is this one called the cricoid cartilage. You can see it here, right? And then there are these C-shaped cartilage rings throughout, as you can see, that prevent your trachea from closing down. Every time you breathe out, they prevent your trachea from completely collapsing. All right, they keep them open. What else do you have? You have these other cartilages like the cuneiform, the corniculate, and the arytenoid cartilage. All right, so these are the other three types of cartilages that keep your larynx open and in the proper shape. The top of the larynx, you have this hyoid bone. The hyoid bone also attaches to tongue muscles, right? So when you speak, the entire apparatus moves. Here's the hyoid bone, also an attachment for your tongue muscles. Look at the C-shaped cartilage posteriorly in the back. These C-shaped cartilages, they kind of keep your trachea open, patent, when you breathe out. So here are your vocal cords. You can see that. your vocal cords, when they are closed, you're silent, not speaking. And this is when you're talking. So your vocal cords are kind of vibrating. That's what makes sound. All right. So talking, vocal cords while you talk, vocal cords while you are silent. Same thing here. When you're not talking, here's the vocal cords, and they're open and vibrating like curtains, right? And that's what produces sound. You can also see the epiglottis at the top. The epiglottis is the flap of cartilaginous tissue that prevents food from going down your trachea when you swallow. So when you talk about the sound, you look at the range, the pitch, and the loudness of the sound. The range of the sound depends on the length and the thickness of your vocal cords, okay? For example, males have thicker vocal cords, so they don't vibrate as vigorously as females, so they have a lower pitched voice. In females, the pitch is quite high because their vocal cords are smaller, more fine, more delicate, so they vibrate faster, right? Loudness depends on how much lung power you have, the amount of air that you can pass across vocal cords, such as if you're a hard rock or heavy metal singer, you really need really good lung power to get that type of a range and loudness. So laryngitis is an inflammation of your larynx that can happen because of too much shouting, hoarseness, because of viral infections, bacterial infections, because of even allergies. All right, any or all of those can give rise to uh, inflammation of the larynx or laryngitis. That might cause your voice to disappear, right? Either partially or completely. Uh, in some cases, this can be dangerous when it can also extend all the way down to your epiglottis, causing epiglottitis. And this can cause sudden airway obstruction and death because of respiratory failure. For example, in kids, um, they might suffer from what is called croup, C-R-O-U-P. And a croup is a lower respiratory tract infection caused by many different viruses, cold viruses, including coronavirus in some cases, and that gives you a characteristic barking cough. When they cough, these kids, it sounds like like they're barking, a barking cough. And croup is actually laryngotracheobronchitis. So your larynx, your trachea, and even your bronchi are all inflamed here. Again, exposure to cold air and cold viruses, all right? That's what it is. Laryngotracheobronchitis called croup. How would you treat this condition? Again, antibiotics. Well, if the kid is young, then make sure that the airway is open. Sometimes they might even need steroids to minimize inflammation, and antibiotic course, and humidification, right? A humidifier to keep the air humid. Okay, so here we are looking at the trachea. So what is the trachea? The windpipe. So again, here's your larynx, the tracheal cartilage, or the thyroid cartilage, or the Adam's apple right in the front. Here's the cricoid cartilage. Here are the C-shaped cartilages of the trachea. Trachea is this windpipe that you can see, okay? The trachea then divides into a right and the left bronchus at a position, at a portion called the carina. This is the carina, okay? You see them dividing into two right here. And this is what the trachea looks like under the microscope. What you see is the lumen of the trachea, and there's these little hair, cilia. Because remember, the epithelium of the trachea is pseudostratified ciliated columnar epithelium. So you see that type of an epithelium right here, ciliated epithelium, okay? You also see the trachealis muscle, which helps you with coughing. It helps again, your trachea from collapsing. It prevents collapsing of your trachea when you cough or when you breathe out. So all of those structures are visible here. You can also see the esophagus, your food tube, which is posterior on the back of the air tube, which is the trachea here. So these are the four layers of your tracheal wall, as seen on histology. Uh, you have the mucosa, which is pseudostratified ciliated columnar epithelium. A lot of mucus produced here in your tracheal region. Submucosa, which is the connective tissue right underneath it, which is rich with blood vessels and nerves. Then the tracheal cartilage, okay? And then the adventitia, which is the outer elastic connective tissue. So these four layers form the wall of your trachea, okay? All right, so then getting down to your left and right bronchi. So here's your right bronchus, here's the left bronchus. You can see that the right bronchus is a little wider and less of an angle. So let's say if a child swallows a penny or something, and that penny gets into their breathing tube, into the trachea, and then goes all the way down. Usually it gets stuck here at the carina, right? Or it's more likely to pass down here into the right bronchus, simply because of the anatomy. You see how it's wider and less of an angle than the left one. So most foreign objects get lodged here. So what will you do in such cases when there is someone who's choking a child who was playing with the penny and is turning blue in the face? You know, the child is in respiratory distress. uh, so first you will try to do a digital sweep, like have the child open the mouth and try to sweep out the stuff. Maybe it's just at the back of the throat and you can sweep it out. Sometimes just like thumping him on the back and they might cough it out, right? But if it's deep down, all the way down to the carina or into the bronchus, then you might have to do a tracheotomy. What is a tracheotomy procedure? As mentioned, tracheotomy or tracheostomy. Where was it? I saw that somewhere. Tracheostomy. Hey, there you go, tracheotomy. So that literally means cutting into the trachea, and that's exactly what you do. So you take a sharp object. If nothing else is available, you can take like a pen or a pencil with a sharp edge, sharp tip, and you just feel for here's uh, the Adam's apple, the thyroid cartilage, just about 4 to 5 centimeters below here. Here's your trachea. Jab it hard. You have to be a little brave in these conditions, right? Don't worry about the bleeding that will happen, but just by jabbing here, what you've done is you've created a hole, or in other words, a third nostril for for the kid to breathe. And you call 911 and the paramedics are hopefully on the way there, right? So what you will hear is, as soon as you create that hole in the trachea, a whooshing sound, because the child is now breathing in through that hole in the trachea, right? So you have bypassed the obstruction now. You've made a temporary opening here till the paramedics get on the scene and they put in an endotracheal tube and start ventilating the child. Tracheotomy might be life-saving under those conditions, okay? All right, so then here's the main bronchi in green. They break down into lobar bronchi in blue, which get broken down into segmental bronchi, even smaller, and then the smaller bronchi, which is in red. So smaller and smaller branches of airways that you can see. Bronchitis of two types, acute versus chronic bronchitis. What's the difference? If it lasts less than 2 weeks, uh, cough, fever, a lot of mucus production, that's acute bronchitis. If it lasts for greater than 3 months, okay, that's chronic bronchitis. Now remember, chronic bronchitis, along with something called emphysema, are the two things that make up what is collectively called uh, COPD, for chronic obstructive pulmonary disease, chronic bronchitis and emphysema. These two disease entities together are referred to as COPD, for chronic obstructive pulmonary disease. It's the fifth leading cause of death in the US patient population, chronic obstructive pulmonary disease. 80% of these cases are caused because of smoking and vaping. Smoking and vaping are responsible for 80% of cases of chronic bronchitis and emphysema, okay? Tremendous mortality and morbidity that happens because of it. Okay, so here you see your bronchial tree, right? All the way down to your bronchioles, and then your respiratory and terminal bronchioles. Okay, so what you see is there is cartilage all the way down till your smaller bronchi. By the time you get to the bronchioles, there is no cartilage there. Instead, you have smooth muscle. You have smooth muscle in your bronchioles, all right? So what that means is if you have uh, asthma, what is asthma? When your smaller airway tracts, like these ones, your bronchioles hyper react, over exaggerated reaction to like cold or pollen or stress or whatever. And so when that happens, when you have an asthmatic attack, these smooth muscles, they contract and then they cause bronchoconstriction. They constrict, they close down your bronchi, your bronchioles, so you can breathe. So that's why you need to give them inhalers, right? And albuterol inhaler, right? Or ipratropium, that's a cholinergic agent. So these types of inhalers will cause or you can give them adrenaline. If nothing else, coffee helps because coffee also causes adrenaline secretion. And so those agents will cause bronchodilation, will cause their airways to open up like this, from this to this. Okay. So there it is, asthma. What are some of the signs and symptoms? Wheezing, right? Some uh, people will not have any wheezing, all right, especially in adults. So that is something called the cough variant asthma. They have this chronic nagging, wet, productive cough, but no wheezing. So that could be a type of asthma as well. It's just there's cough, no wheezing there, okay? So bronchoconstriction, wheezing, coughing, excess mucus, uh, and this is because of a localized immune reaction, right? Again, it's autoimmune. Even asthma has an autoimmune component. Your immune cells are reacting to your bronchi and your bronchioles. All right, how you treat it is with steroids because steroids, remember, dampen inflammation and bronchodilators, okay? So here are your alveoli, these grape-shaped structures, right? And what alveoli do is they increase the surface area available for the exchange of gases, right? So your oxygen and your carbon dioxide get exchanged here. It's a very vascular area to see. There's a number of blood vessels all around your alveoli because gas exchange is taking place here from your lungs uh, to your blood circulation. So here, this is what actual alveoli look like under the microscope, right? Here's the respiratory bronchiole, these little grape-like spaces, spongy tissue here. Those are the alveoli, okay? So here are the different types of cells that you will find in the region of your alveoli, right? So the types of cells that you find within the alveolar walls is what we are looking at here. Um, so alveolar type 1 cells, 95% of your alveoli are filled up with these types of cells, okay, and they simply form the barrier, the blood gas barrier in between the actual bloodstream and the lung epithelium. Then you have the alveolar type 2 cells, okay, and they are found within the septa. Septa are the dividing tissue in between the different alveoli. And they basically are extremely important because they secrete an oily secretion called surfactant. And what this oily surfactant does is it prevents the alveoli from sticking together, right? In other words, they reduce surface tension. Surface tension is a characteristic of water that allows different wet surfaces to stick together, right? Think of trying to blow up a wet balloon versus a dry balloon. So which one would be easier? Of course, it's much easier to blow up a dry balloon as opposed to wet because the wet balloon is going to basically stick together. Same with the lungs, right? So the lungs are wet on the inside, and so that uh, would cause us to exert a lot of effort just trying to breathe in because of the wet lungs basically sticking together. But what surfactant does is it prevents them uh, from being stuck, right? So therefore it reduces surface tension. And then finally, dust cells, also called alveolar macrophages, and they literally are the Pac-Man cells of the lungs. They eat up all of this little debris and dust that might be in the lungs, okay? Okay, so here you're looking at your alveoli, of course, zoomed into those under the microscope, and you can see the different layers that are found within the respiratory epithelium, right? You can see the oxygen moving in and the carbon dioxide moving out. Here's your red blood cell. Here's the alveolar epithelium itself, right? Here's the fused basement membrane of the alveolar epithelium and the capillary, and then finally the alveolus. So the gases move across these three layers here. So how does the larynx assist in increasing abdominal pressure? Well, the Valsalva maneuver, right? Whenever you cough or strain, the larynx actually closes, and that causes your intra-abdominal pressure to increase. What are the three unpaired cartilages in the larynx? The cuneiform, the arytenoid, and the cornicuate cartilages. What are the structural and functional differences between the vocal folds and the vestibular folds? The vocal folds are the true vocal cords. Vestibular folds are the coverings in front of them. What is the function of the C-shaped tracheal cartilages to keep your trachea open when you breathe out? to keep them patent. How do the trachealis muscle and elastic ligamentous membrane that complete each uh, how do the trachealis muscle and elastic ligamentous membrane that complete each ring posteriorly function? So again, they keep your trachea open or patent, okay? What are the significant structural differences between bronchi and bronchioles? Bronchi have cartilage, bronchioles do not have any cartilage. Instead, they have smooth muscle. Which of the following respiratory structures is supported by cartilage? Nose, yes. Larynx, yes. Trachea, yes. Bronchi, yes. Bronchioles and alveolar sacs, no. All right, the respiratory tract can be damaged from drying out or desiccation, cold air, microbes, or exposure to chemicals or particulate matter. Which of the following helps protect the respiratory tract? Nasal hairs, they do. Mucus, yes. Tonsils, no, because they're at the back. Cilia, yes. Macrophages, yes. Sneezing, coughing, yes. So except for tonsils, all of them apply. All right, list the conducting and respiratory structures in order that air passes through from the atmosphere to the alveoli. So first through the nose, then the paranasal sinuses, then the pharynx, then the larynx, then the trachea, then the two bronchi, then smaller bronchi, then bronchioles, and then to the alveoli. List in order the structures of the respiratory membrane that oxygen must cross to move from an alveolus into the blood. From the fused basement membranes of the epithelium of the alveoli and the uh, capillary, then the alveolar epithelium, and then into the alveolus itself. So here we are looking at the lungs themselves, the right and the left lungs, all right? So what you can see is they are located on the other side of the heart. Here is the heart in the mediastinum, the middle of the chest, the two lungs on either side. The top portion here is called the apex of the lungs. The lower portion here is called the base. The base of the lungs rests on the diaphragm. Here's the diaphragm, the muscle that separates your thorax from your abdomen, okay? What else can you see? You can also see that the left lung is kind of collapsed here. This is called the cardiac um, notch, the cardiac notch, because the heart basically fits here into this notch, okay, on the left-hand side. You also see that the right lung has two fissures, right? One and two. So as compared to one transverse fissure and one oblique fissure here. If you look at the left one, there's only a single oblique fissure. So the right lung is divided into an upper, a middle, and a lower lobe, while the left lung is divided simply into a superior and an inferior, an upper and a lower lobe. So all of these structures are visible, okay? So the horizontal fissure uh, in the right lung, of course, separates it into a superior and a middle lobe. The oblique fissure separates middle from the inferior. The left lung is smaller. It has a cardiac notch, remember, to fit in the heart. And there it is, the cardiac notch. And it only has a single oblique fissure, which separates its superior and inferior lobes, okay? All right, so here you can see the difference between the two lungs, the horizontal fissure and then the oblique fissure in the right lung, therefore the superior, middle, and inferior lobes, and a single oblique fissure in the left lung, so it's only a superior and a inferior lobe. This is called the hilum or the hilus of the lung, and this is the region through which the bronchi, the pulmonary arteries, and the pulmonary veins emerge and exit the lungs. So you can see here again the right and the left lungs, they are divided into different segments. The segments are colored differently. The importance of this structure, or rather this picture, is this shows you that sometimes when you have to cut out pieces of the lung in someone who might be suffering from like advanced tuberculosis or anthrax or even lung cancer, you can cut out certain segments from here, okay, while leaving behind the others, and they will basically take up the function of the missing lobes. Now remember, you can donate safely one lung and function on the other one, right? You're a little bit more vulnerable to infections, but in paired organs, you can donate one of the pair, then you can still survive, like your kidneys. You can donate one kidney, the other kidney will take over the function. Same with the lungs, all right? So here what we are looking at is the cause for 80% of lung cancers and COPD, smoking itself, all right? And this includes vaping as well. So even though vaping has a slightly lower risk than actual cigarette or cigar smoke, but it does have substantially higher risk than someone who doesn't smoke at all. And this is only a partial list of what can go wrong with someone who smokes, all right? So everything from COPD. We talked about how 80% of chronic bronchitis and emphysema has to do with smoking, to atherosclerosis, right? Fatty plaques forming in your arteries, to lower birth weight in pregnant women, uh, ear infections, lung infections, of course, and the worst of all, lung cancer, 80% of which can directly be related back to smoking. So if anyone needs any more reasons uh, to to quit smoking or not to start, even better in the first place, but this is only a partial list. There's many more reasons. So blood supply to the lungs, as you can see here, you have both the pulmonary and the bronchial circulation, okay? So the bronchial arteries, which branch off from the descending aorta, and the bronchial veins drain back into the pulmonary veins, okay? Here in this picture, you can look at the aorta giving out the bronchial arterial branches, the bronchial arteries, and then the bronchial veins draining back. Um, and eventually they'll all, and the pulmonary veins will drain back into the superior vena cava. So lung cancer, now this is by far the top killer of all cancer types in the world for both men and women. Earlier, men used to have a higher incidence of lung cancer because they used to smoke more. Now it's an equal opportunity killer. So males and females are equally susceptible to death from lung cancer. One of the most aggressive types uh, of cancer. Smoking is responsible for 85% of cases, okay? Some signs and symptoms, weight loss, chest pain, um, chronic cough, or you might have none of those, just like vague signs and symptoms, fever of unknown origin, those kinds of things. But it kills more people, as I mentioned, than any other cancer, okay? So the three types: squamous cell carcinoma, and it's called squamous cell because it arises from uh, pseudostratified columnar epithelium actually changing into a squamous cell type epithelium, okay? It happens in smokers because during ANP1, we talked about something called metaplasia. Metaplasia is when one type of epithelium changes into a different type because of chronic insult or damage. So this is what's happening in chronic smokers. They keep smoking, and the smoke damages the pseudostratified columnar epithelium of their trachea, bronchi, which metaplasias, undergoes metaplasia and turns into squamous epithelium, stratified squamous epithelium. This is the first sign of damage. If the person does not cease smoking at that point, then this squamous epithelium will turn into carcinoma. So that's squamous cell carcinoma, as you can see. Adenocarcinoma, it arises from the glands, the mucus producing glands in the lungs. And finally, the small cell carcinoma of the lungs, which is produced by the neuroendocrine cells in the bronchi themselves. Regardless of the type of cancer, the treatment is the same: chemotherapy, surgery, and radiotherapy, okay, depending on the stages. But again, lung cancer is one of the most aggressive malignancies out there. So do not smoke, avoid smoking, right? That's the main thing. Clean air, stay away from pollution. Vaping is included in this whole picture. So these are some of the things. Also, if people in your family had lung cancers, then you have to be on the lookout a little bit more. There is a genetic component, pretty much like all other cancers here as well. Okay, so the respiratory system is innervated by sympathetic nerves from your first thoracic to the fifth thoracic regions, okay? And what the sympathetic innervation does is it causes bronchodilation. So your sympathetic nervous system is your fight or flight mechanism, right? It releases adrenaline, and that's why you drink coffee or take adrenaline shots when you're having asthma, because they will help you bronchodilate. Parasympathetic nerves come from the vagus, which is your 10th cranial nerve, and this causes bronchoconstriction. Your lungs collapse, they close down here actually, okay? Okay, so pleura are the serous membranes that surround your lungs, right? And we saw those pictures initially. The serous, there's the parietal pleura and the serous, the visceral pleura In between the two, the parietal and the visceral pleura, you have a pleural cavity, which is filled up with some fluid, less than 15 milliliters of fluid. All right. And why do we have this fluid surrounding the lungs? So when you breathe in and breathe out, when the lungs expand and collapse, it prevents friction. So that's why it's there. But sometimes you have infections like pleurisy, which is an actual inflammation of the pleural membrane. Okay. And when that happens, there's inflammation. And if you listen to their lungs, these people with pleurisy, they complain of chest pains while breathing. But even when you take a listen with a stethoscope, you'll hear this rustling sound, and that's called a friction rub. All right. So in pleurisy, you will pick up a friction rub on auscultation. Listening to their lungs with a stethoscope, and you hear this. All right. Pleural effusion is literally excess fluid in the pleural cavity. Why? Because of infections, because of an injury, even radiation exposure to the lungs, uh, liver cirrhosis in chronic alcoholics, someone who drinks a lot of alcohol, heart failure, lung cancer. So we have a long list of conditions that can give rise to pleural effusion. On a chest x-ray, if you see fluid in the lungs, you have to basically narrow down the pathology. What is it exactly that's causing this pleural effusion there, all right? What's the treatment? In any case, if you have fluid around the lungs, you have to put in a chest tube. You have to put in a chest tube, and then you have to drain out the fluid through the chest tube from there so the person can breathe without any problem. All right, so there you see, here's those are the pleura. That's the parietal pleura on the outside. Here is the visceral pleura on the inside. In between the parietal and the visceral pleura, you have intrapleural fluid, right? Or pleural fluid here, as you can see. The diaphragm, the muscle that separates the chest from the abdomen, you can see that here as well. Pneumothorax is another condition where what happens is, let's say somebody gets stabbed with a knife or a sharp object in the chest, or gets shot, and this would actually puncture the lung, right? There's a hole in the lung now. So every time you breathe in, this air from your lung is actually going to escape out into your chest, all right? But the chest is it's a closed cavity. So with each breath, you're pumping out fluid out of your lungs, not fluid, but air out of your lungs. And this air is gathering up in your confined chest cavity, and your chest can only swell so much. So what does this air do? It presses inwards, causing lung collapse. That's what makes, and this is called a tension pneumothorax, okay? And that's what makes pneumothorax such a dangerous condition, all right? Tension pneumothorax. So how would you treat someone with a gunshot or a stab wound to the chest who's in pneumothorax or tension pneumothorax, right? Where air, with each breath, air is escaping from the ruptured lung and accumulating in the chest cavity and then putting pressure on the lungs and collapsing him. You need a chest tube again. You need to put a chest tube, and you have to, this time, drain out not the fluid, but the air itself, all right? And you have to surgically patch up the hole in the lung as well, so that every time that the person breathes in, this air doesn't escape into the chest cavity and cause pneumothorax. Atelectasis means a collapsed lung, all right? And again, this happens with tension pneumothorax, right? All the air on the outside will cause your lungs to collapse. Treatment, again, is a chest tube. You have to put that in. All right, so which arteries deliver oxygenated blood to tissues of the lungs? Bronchial arteries, pulmonary arteries. And which veins drain deoxygenated blood from the lungs? Bronchial veins, pulmonary veins. What vessels receive some of this deoxygenated blood? The pulmonary veins, again. Well, pulmonary veins actually carry oxygenated blood, so pulmonary arteries. What is the function of serous fluid within the pleural cavity? To prevent friction every time the lungs expand and contract. Why is the intrapleural pressure normally lower than intrapulmonary pressure? So that the lungs stay inflated. You don't want to push the lungs inwards for them to collapse. That would be tension pneumothorax. What is the function of this difference in pressure? To keep the lungs inflated and not collapsed. Okay, so that is it, the end of chapters 22 and part 1 of 23. Okay, so we are looking ahead at our spring break for this week. So no lectures, no recordings will be sent out on Saturday. Um, when we meet again on the Saturday after, that is the 27th of March, we're going to have our lecture exam number 2. I have sent out the instructions for that. You also have the case studies guidelines, so please work on those, and we will pick up the labs from that point onwards. Okay, so have a wonderful, fantastic, relaxing, well-earned spring break, at least from college classes. Try to get your Mediterranean diet and your exercise and your stress and sleep schedule going. All right, now is a good time as any to start, and have a wonderful time. Stay safe, as I said, and I'll see you all in a couple of weeks from now. If you have any questions in the meantime, suggestions, feedback, feel free to let me know. Bye-bye.